The other one is I didn't mean to cause this problem with the file naming of the protein files not lining up with the names that were on the slides. But in some ways, it's a good learning lesson in that when you are putting file names in the Slurm script and you go to the error file because it didn't finish and it says file not found, that's almost a sure indicator that either your path is wrong, like those data files are not in the directory with the Slurm script, or you have a typo in either the Slurm script where it named the file or the file names are not what you think. Those two are not matching. Those two things, so the command not found because the typos in the job script name and typos in the file names are all of the problems that I've seen personally. So that's logistical stuff about being able to get it done. I glossed over the reciprocal best blast because you really don't have to code this. If you give ChatGPT these two files, for example, the outputs of, in this case, TCAS versus AGLA and AGLA versus TCAS, and tell it to give you the reciprocal best matches, it's pretty smart. It will do that. And then given the TCAS AGLA overlap, so you get one file back for that. And so that would be this file on the left. If you do that for all three of them and then ask ChatGPT to give you the overlaps for all three of those sets, one case where it gave a blank file. So in previous years, if you go through the code that it gave you to generate that, or ask ChatGPT why is the file blank, it can usually figure out why. And it's because the column order is not allowing it to go in a circle. So for example, if AGLA and LDEC were flipped here, the way the dictionaries are checking is it says, okay, is this accession number for AGLA, get that TCAS number. So now I've got TCAS number. Now go look in this next dictionary and find that TCAS number, get the LDEC number that corresponds, then go to this dictionary and find that LDEC number. Well, if these two columns are flip-flopped, it can't find that LDEC number because it's not a key in this dictionary, right? And so a way to solve that problem easily for ChatGPT is to name the columns. So if it has species headers, ChatGPT is pretty smart and it will recognize that. At least from my experience, it recognizes the headers and it will use those to do the intersections. But you can also tell it to sort of disregard that kind of information and just tell it what the potential problem is, and it will fix it itself in its code. Very useful for you to look at that code and try to understand what it is. So that's just a way to reinforce Python knowledge as well. Any questions about that? Okay. How many of you have asked ChatGPT why? Not that many. More people should ask why. I've told a few people this already, but like if we get to the end of the semester and you say, what did I need Dr. Damuth for? I just had ChatGPT. that's a win for me. Like, that's the goal. Uh, Learning how to use it and asking why is just one example. Okay, so that's all the reciprocal best blast, how to find orthologs by that matching across and among species. What you often see in the literature when somebody wants to define orthologs is that they have used OrthoFinder, which over the last 5-10 years has kind of been the de facto. For a while, it was really hard to get set up on local machines because it has lots of dependencies. The Colab notebook that I linked to in the Canvas module, at least for me, it's just you can run the whole notebook and it will do all the OrthoFinder steps as long as you've got the data in the right place. What OrthoFinder does, first, well, there's been multiple revisions. Originally, OrthoFinder just did clustering based on similarity, and that doesn't define one-to-one orthologs. It defines clusters of orthogroups. So it uses an algorithm called Markov clustering. The graphs over here on the right, what you can think of is every node in this graph is a gene, and every edge is the similarity score. And so what ends up happening across iterations of the MCL is that you start to see clumps of genes that have short edges, right? The genes that are most similar to each other have the shortest edges. And it is disregarding which species the gene came from. All it has is just a giant list of gene sequences, and it says, you know, do the fastest algorithms around, just break it into k-mers, and then say, what's the k-mer distance between these two sequences? And so it builds those edges based on that score. Now, MCL is a tunable algorithm. So you can say, what I mean by tunable. You can weight the edges by some constant, or say by a log score, so that things that are close together are extra close together and things that are farther more distant. So it's a way to try to expand the graph, but not necessarily in a linear way. So that's what I mean by tunable. If you want to have families that are more inclusive, you can tune it to be more clumpy. And if you want it to have gene families that are closer to one-to-one every time, then you tune it so that it splits at lower values for that distance matrix, or lower values. So, concrete example. I could say split at 99% similarity, right? In which case, there would be very few genes that are present in all species, okay? Because what it's doing is splitting. If it's more than 1% different, it's calling it a different family, okay? So what ends up happening then is your gene families look like present, present, absent, 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 right? Because all those things that were more distant, species that were more distantly related, the copies of that ortholog are more than 1% different, just because the species have an old split. On the other end of the extreme, I could say everything that is 1% different is in the same family, or greater than 1% different. So now everything belongs to one family, because that's like has almost zero similarity, but you still want to count it as the same family. This is a silly example, but you see the ends of the extreme, right? You're calling individual genes as their own family versus you're calling everything as one family. This is a continuum. Like, what do you use? That is, most people just use the default. I think it's something like, I forget what it is now, 92%, something like that, on average. So that's what we mean by tunable. You can clump the families more or split the families more. That's still the first step in OrthoFinder. OrthoFinder 2, which came out 2017, where there were a couple algorithms, and then 2019 is really the full revision where they implemented it with MCL clustering. What this does is take each of those MCL clusters and build gene trees, rooted gene trees. So it's trying to plot, right? So go back here, right? So the difference between a gene tree and a species tree. So in my example here, the only way you know that this homolog got lost in species 2 is by reconciling the gene tree with the species tree. So you say, well, this B copy, so there was a duplication down here that we infer, and this is the gene tree. So it says 3B and 1B are the most similar to each other, and all the A copies are similar to each other. Well, the most parsimonious explanation for how this happened, if you overlay it on the species tree, is that there was one single loss of that 2B copy. So this is what OrthoFinder 2 did. It said for each gene family, do this species tree, gene tree reconciliation to try to identify where the duplications happened and where the losses happened. And then the last step was to integrate some iteration on those species trees to try to figure out confidence. But really, OrthoFinder 3 was about making it faster. And so in terms of the algorithms themselves, the species tree, gene tree reconciliation is still the main benefit of using OrthoFinder. So this is still preprint. Version 3 is not published in peer review yet. But the main points are just that it's faster and more accurate. And that's partly to do with this iteration at the end. The accuracy is. Like I said, it hasn't in the past been trivial. It's still not trivial to get working on an HPC. That's why I didn't do OrthoFinder on bridges. And then the flip side is we could have done it in Colab, but it takes 5.5 hours just on those 2 sets that we did. So luckily you've got all the time you want on Colab. I guess there is a monthly cap, but probably nobody's going to run up against that. So you could run that Colab notebook if you want to. It's not eating into our course allocation or anything. If you just want to see, well, really you just see the little play wheel spinning for a long, long time while it waits to run the next steps. But those outputs are Canvas. Right, so there are more than just these 2 folders, but the orthogroup folder and the comparative genomic statistics folder have all the outputs. You actually don't need all those outputs. I put them both there just so you'd have to look at them. Give those to ChatGPT and ask for the 3-way single copy orthologs, and ChatGPT is smart enough to know. Well, tell it this is OrthoFinder output, how to give you that number. Anybody finished that yet? I haven't gone and looked at the quizzes. You got it? So more or less orthofinder three ways than reciprocal best blast three ways. You remember? Which so which one do you get more of for anybody who's finished the orthofinder part? Are there more three-way single copy orthologs in the reciprocal best blast results or in the orthofinder results? Yeah, there are way more in the reciprocal best blast results. And so, go back to the PowerPoint here. For, let's see, I can't blow that up any more than that. But at the end, another new thing for OrthoFinder 3 is that it outputs these things called hierarchical ortho groups. And so N0 are the ortho groups that it infers to exist at the root of the tree. So in our case, these three beetles share a common ancestor. It will only call a three-way ortholog if it infers that it was present at the base of the tree. And so that's one one place where it could be more conservative, so it's giving you less. The other one, and probably the one where it's losing the most, is if it has one in TCAS, one in AGLA, and multiple in LDEC. It doesn't do any more work to try to figure out which one in LDEC match should go with those other two. Whereas in our reciprocal best blast, if there's a single LDEC copy in there that works in that three-way reciprocal algorithm, it's still going to give us that family as one of our ortho groups. Does that make sense? This is probably a lot to digest on the fly, but if you wanted to bring those numbers more in line, what is something you might try? So if we go back to here, if one of the main reasons that OrthoFinder, that example of 1, 1 multiple, or say multiple, 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 like there's 3 in each, and OrthoFinder says, I can't decide, I'm not even going to put in the effort to try to figure out which one's orthologs, you could tune the MCL to split more. Okay, so some of those families that have multiple in one species now don't have multiple, and that potentially will increase the number of one-way or three-way single copy orthologs. Now, you don't have to do all that, but this is a real biological problem that some of you in the course of research may come across. Gene family evolution is probably what got me this job. I helped write in my postdoc lab a software called CAFE, which is analysis of gene family evolution over phylogenetic trees. And so I probably, like, I could get lost in the weeds for another hour on this stuff. But this is a very common problem because all of evolutionary inference depends on you comparing orthologs. When we started out for CAFE, it wasn't because we were interested in gene families. It's because we were having such a terrible time trying to figure out which pairs of genes were orthologous to each other between human and chimp. It's like we got 5 copies in human and 5 copies in chimp. I don't know how to do evolution on 5 against 5. I can only do evolution on 1 against 1. Okay? All right, so I'm not going to say any more about that unless there are questions. Take 5 minutes. Adam and Zoe can help go around if you're having trouble with your blast. Okay? If you're already set, everything's working, move on, go get the data for the next module. So module 7, the PowerPoint and the data are already up there. Anybody over here having trouble with?
I do have a question though. When
Yep.
you mention it being tunable, if you're doing like the beetles that you do, can you put in only things that you already expect to have orthologs? Like, because the picture had like the dolphin and like the mammals and stuff. Would that be a way to kind of?
Everything has orthologs. The question is how many.
Okay.
So if you have things that are really closely related to each other, you can make the clustering part, that tunable part, you can set it at a higher threshold. So if I know my species are closely related, I could make it 95% instead of 90% as the cutoff. Mm-hmm. With and without duplicates, yes. So I forgot your name again. Elaine. Just brought up a question that's come up a few times. So in the one-way blast, there are duplicate lines. Why? Because we told it to only keep the best hit. But there are duplicates in there. Any guess as to why? It keeps all the maximal segment pairs for the same genes. So it's only keeping the best gene, but if there are 2 sections that match for that gene, it gives you 2 lines in the blast output. Now, the quiz will take both answers. So if you remove the duplicates or if you keep the duplicates in there, it's going to count them both correct. But it's something to be aware of. Log that in the back of your head for some point. Mm-hmm. Mm-hmm.
So I actually run the whole process, and the 3 ortholog for the reciprocal blast is actually less than the also find the analysis. Is that right, or is it
that 3-way ortholog? group for OrthoFinder is too high, I think.
This one is too
Yeah,
high?
I don't think that number is right.
So it should be like way more lesser than the reciprocal blast.
Mhm.
I'll just write it again.
So you you shouldn't need code to get this answer. It's output in, like, it tells you in the comparative genomic statistics.
Also, is it right to use this file, right?
Yeah. Yeah, so this number of, so you took that number.
Yeah, I guess so.
So that's number of ortho groups with all species present, but we want all the ones that are single copy, right? So in here, you've got families that are 1, 3, 5, and 10, 50, 25. You want only the families that are 1, 1, 1. And so that's the number of single copy ortho groups.
This one?
Yes. So that's what ChatGPT would have told you. So I just saved you having to type that into ChatGPT.
Thank you.
Jeff, do you know if there's like a loss and found for this building?
No idea.
No, no, no. I don't even know who to ask about that here.
UTA police. Yeah.
But didn't you say in the vaccine earlier that the number of the orthologs, assuming ortholog groups from the orthofind analysis should be less than the one from the reciprocal blast?
Yeah, but that one's way too low. Actually, that number, I believe, is too low.
So the problem starts from here. It's kind of strange that when I actually did this calculation, it's way more less than the other answers.
That number is wrong. Right. So I wonder if this job didn't finish.
So what kind of job?
The blast job. Maybe it didn't finish.
Well, I may not receive like full complete sets before I run the codes, but maybe I should just like try to just do reciprocal as all over.
So how did you get that number?
Well, I just use the old code I use for all the questions.
Uh-huh.
Yeah.
Well, except these don't have FASTA headers like those did. Like, so I don't know how you did the first one, but the first, like, to figure out how many proteins are in there, you count the number of FASTA headers with grep or something like that. For these, you're just counting the number of lines in the file, right?
Yes.
And so that there have to be way more lines than that in the file. Actually, these numbers also look too high. Because, so go back up. If there are only 20,000 genes, you can't have more blast hits than that.
This is a problem of the header.
I guess in principle you could have more, but you shouldn't. I'm just not sure how you're counting. For the blast hits, you should just count the rows, and for the genes, you should count the FASTA headers.
Oh, I actually didn't get this part. Like for the gene, they should just count the header.
Yeah, so you know how the FASTA file has header and then sequence?
Oh yeah.
So if you just grep on that and do grep-c, it will count. So that tells you how many headers, that tells you how many proteins are in there.
Oh yeah, I think this is the code I used.
Yeah. And then for the blast hits, you've got XP and then some number, and then maybe NP and some number. There's two columns. And so the number of rows in that file is how many blast hits there were.
Oh yeah, that's true.
Yeah, so it does. Some of them look like they're in the right ballpark, but not all of them. So if it's the same code, they may not. It may be that none of them are right. I don't remember the numbers off the top of my head, but office hours, yeah. Or if we get through stuff, then maybe there's time at the end. Okay, I'm going to move on to genome variant analysis. Yep. Yes. I'm going to try to fit it back in later, but yeah. Okay, so we're, roughly halfway through the semester, so just wanted to kind of take a second to say the overview in the background there. Can you see the forest with the tree blocking the way? Because like the tree is, I can't get my blast to work, but I don't know why I'm trying to get my blast to work in the first place. So we started out with Unix and Python, just very cursory basic stuff, but then we got into the pattern matching stuff with Rosalind. So we learned to take short sequences and try to find matches in the genome. Then we sort of relaxed the matching rules, and with the biological question being, can we find first the origin of replication? Okay, and we brought in the GC content, some biological information to help us make that a computational question. Once we figured out how to relax the pattern matching, then showed how you could use that to find transcription factor binding sites, for example. Things where, if you remember those sequence motifs, where you've got a higher probability of a nucleotide being at a certain position. But that algorithm, that search strategy, works fine for those kinds of questions. Then we talked about alignment. So when you're doing pairwise alignment, spent a whole lecture, didn't really mean to, but getting to why brute force and greedy algorithms either don't work or aren't practical because it's just too big a computational problem, and how the computer scientists solved this problem with dynamic programming. So it will give you the optimal alignment between two sequences. When you extend that to multiple sequence alignment, the main algorithms that help get to the best alignment for multiple sequences were the progressive and iterative algorithms, where right, it's building a consensus as it works its way down, so it's always doing the optimal alignment between two sequences. Not necessarily guaranteed to give you, especially if you've got really big sets of sequences, the optimal alignment among all of them, because it depends at each step on which pairs it tried to align. Talked about local versus global alignment, which is connected to how you score gaps, especially like what's the cost of inserting a gap versus extending a gap. Then this past week we talked about BLAST and how if you want to search, if you have a specific sequence and you want to search a big database, that pairwise multiple sequence alignment strategies don't work. And so we have this heuristic search method, and I don't call it an alignment method, even though it is doing some aligning, but it's not guaranteed to give you the optimal alignment. It's just guaranteed to find similar sequences. But that's what you have to do when you want to search big databases. You do the alignments after that. And we showed how you can use that. It's what you were just working on, you can use that sort of strategy to define important biological questions about what is homologous, orthologs and paralogs. So looking ahead, what we're trying to get to, today we're going to start genome variant analysis. So what happens if you've got a reference genome and you do some resequencing? How can we figure out where the mutations are or where the variants are? And then hopefully, depending on where we get today, either Thursday or next week, we'll do genome-wide association. So how do you make that connection between genotypes and phenotypes? When we finish that, we'll do transcriptomics, and that will lead us into machine learning and using classifiers to try to figure out either which group of individuals. So we'll do classification based on gene expression profiles. We'll also do classification based on species abundance, so like metagenomic clustering. Does the microbiome differ between habitat A and B or host A and B, for example? And then hopefully we'll get to single-cell RNA-seq and metagenomics before you get turned loose on individual projects. So today, genome variant analysis. The biological questions here is pretty fundamental. Is there a genetic basis for phenotype X? The computational challenges that come along with that are read mapping, variant calling, and then making the association. I'm hoping today we can get all the way to a VCF file, a variant call file. Now, even though I'm going to talk about this, the sort of trajectory of this tutorial is toward genome-wide association. So the most common context for genome-wide association is human disease mapping or human trait mapping. How do you make the association between human variants and traits? But lots of, especially the PhD students in here, are in labs that are interested in comparative genomics for evolutionary kinds of questions. And so certainly up through variant calling, it's the same pipeline, right? You have to know what the variants are before you can make any evolutionary inferences about how they got to be that way. So looking at nucleotide variation between individuals in a population or between populations or between species, all of those downstream questions, when you take molecular evolution, you sort of learn what to do with the variants if you want to infer something about evolution. And hopefully we'll have some semester projects that use variant analysis to do some comparative genomics, evolutionary genomics. So a couple other ways this tutorial is tailored to emphasize certain things. There are lots of ways we could go, lots of different kinds of data we could use. We're going to use Illumina data because that's the most common platform. It's the lowest cost and highest accuracy per base. So if you're not using Illumina, there's got to be a reason. The default is Illumina. We're going to talk about whole genome resequencing and mapping. We're not going to do de novo sequencing or gene prediction because those are typically sort of one-off projects, unless you're in a comparative genomics lab where you're doing lots of new genomes. But in terms of bioinformatics, what bioinformaticists do, they may do a new genome where, so by de novo sequencing, I mean sequencing a species or an organism that's never been sequenced before and then assembling the genome, then predicting the genes. Usually you do that one time, and then the process of making it better is kind of a forever problem. But that's not what most people spend most of their time doing in bioinformatics. So we're going to not do any of that in class. Again, maybe somebody will have a cool end of semester project that does that. And we'll also talk about assembly. So we're not totally going to skip over how assembly works, because we'll talk about assembling transcriptomes, which is similar. We're not going to use genome-wide SNP genotyping. So if you've ever heard of SNP chips, so especially for humans, all of the common variants in human are known based on population genomic sequencing. And so you can, like, they're tethered on a chip, and you can just look at those variants using the technology is called SNP chips. So in a clinical setting, that's pretty common, right? It just gives you back the readout. Is this person homozygous, heterozygous, at that particular known variant? We could do that, but a couple reasons why not. One, that approach is, by nature, can't discover new variants, okay? So if somebody has a novel mutation, you cannot find it that way. So it's way more common in a clinical setting, where if somebody has a novel variant, it's never been seen before, in a clinical setting, you can't tell them what that does anyway, right? So SNP chips are good for known information and telling people sort of, do you have it or not, based on what we already know. So they're less common in research situations. In research situations, it's almost always whole genome sequencing. The other thing is, even in the clinical setting, whole genome sequencing is becoming more common because the cost has come down so much. So we can do human genomes on the order of about $200 now, which that's not a lot more expensive than doing a SNP chip. Now the bioinformatics cost of doing the whole genome instead of the SNP chip is a little bit higher, but everything is moving in the direction of just do the whole genome and ignore the SNP genotyping. We'll do quality control of the read data, and then identifying variants today, and hopefully make associations on Thursday. Vocabulary slide, because I know I've already been throwing around lots of jargon. There's a link here. That's the video that's already posted in Canvas. So the Illumina workflow, there are multiple Illumina platforms. They're all short read sequencing. So in case you aren't already totally familiar with the verbiage, like, reads are just the chunks of sequence. Paired end sequencing, when we're reading off both ends. I've got slides for this in a minute. And then the read length is just how long are those sequences. What we're going to do today, the read mapping, that's just aligning those figuring out where those little reads belong or where they came from in the genome. Coverage depth, how many times on average have you sequenced a single base pair? So when you do whole genome next-gen sequencing, it's not like you just get a straight readout of the genome, right? You're fragmenting the whole genome into little bits and then sequencing all those little bits. And so because it's random, some little bits you sequence a lot, and some little bits you don't sequence hardly at all. How does Illumina sequencing work? I've given you the video, but it's important as a bioinformatics person to understand how the data is derived, because that tells you something about how to process it, what kinds of errors it might have. This is the example of 4-color chemistry in Illumina. They have 2-color and 1-color now, but it all works essentially the same in this sequencing by synthesis. So it has a glass slide with a template tethered to it. The first step in the library prep is that you put an adapter on your genome that you want to sequence. So first you fragment it all those fragments get a little adapter. That adapter is what anneals to these probes that are on the glass slide. So you've got your template DNA, that's the gray piece sticking up, and then that's tethered to the slide. I'm skipping over several steps. You should watch the video. It's linked again here. Then you put the DNA polymerase in there. It binds to there's a primer site right next to that tethering sequence. And then that thing incorporates one nucleotide, and there's a terminator on the nucleotide so that you can't extend it more than once per round. So it incorporates one nucleotide, takes a picture of the whole slide, and so that's what you're seeing down here. Like it shot it with the laser so that everything on the slide fluoresced, and that tells you what color just got attached to each one of these strings, okay? So the raw data for Illumina sequencing is really just lots and lots of pictures. And so it keeps track of the position in the image so that, for example, here I've got two sequences being built. In round one, both of those two little spots that I've got circled fluoresced green, so we know there's a T there. In round two, this sequence up here fluoresced blue. The one on the bottom fluoresced green, so you add the letter. And so that you just round after round are building the sequence. Okay, that's sequencing by synthesis. Skipping over lots of details, but that's kind of the level just to give you an idea how these things are being generated. Why bioinformatics people, despite ChatGPT, are not going to be in short supply for a while. This not going to be in short supply? No, they're going to be necessary for a while. They are in short supply. Lots of jobs out there for people who know how to deal with this amount of data. Just to give you scale, the human haploid genome, so how many unique nucleotide sites are there in a human genome? The ballpark is 3 billion bases. The NovaSeq X can do 8 terabases in 2 days, right? So thousands of human genomes in a couple days. If you look at their ad on, I want to call it propaganda, on the Illumina site, the NovaSeq X Plus is like 2 of them in the same machine, so it doubles everything. It can do 128 genomes at 30x coverage in one run. So it, in a year, you can. This is why it only costs $200 to do a human genome now, and the accuracy is really high. This is far and away the highest throughput machines. Something I skipped over when I was talking about SNP chips. We're also, we could do exome sequencing. So there are probes for human where you can pull out just the protein coding parts of the genome and just sequence that if you're not interested in the non-coding parts. The pipeline for that is the same, okay? So whether we had exome data or whole genome data doesn't matter. We're going to do E. coli just because it's fast anyway. Another bit of Illumina, need to know. Illumina is a short read technology. So I keep sort of mentioning it as a side. There are lots of other technologies. The main long read technology now is called PacBio. And so it will sequence, instead of a couple hundred base pairs in a read, several thousands of base pairs in a read. Why that's relevant is our computational challenge in mapping is figuring out where did this read come from in the genome. If it's only 100 base pairs long, there are potentially lots of places that match it pretty well, okay? And so trying to decide which place it came from exactly, so mapping it uniquely, is hard if you only have one read, and also the human genome, for example, most multicellular have lots of repeats in them, transposable elements. And so all of those things make it hard to map single end reads. And so fundamentally, this is a problem, just like we started out the semester with, of pattern matching, okay? But it may not be a unique pattern in the genome, and we have millions and millions of those patterns to go match. So paired end reads just give you more information to find the unique spot. Paired end means you're sequencing the few or couple hundred base pairs. In this case, the most common is 150 base pair reads off of both ends. And then there's a gap in the middle that is not sequenced, but you know roughly how long that is. So when you go try to map it to the genome, one of them matches, and then if you don't find the other end of that sequence in that sort of range of how big the fragment is, that's probably not the right spot, okay? So it helps you find the right spot. Paired ends, and then also the longer reads. Like if you've got PacBio reads, it's much easier when you've got 1,000 base pairs because you're less likely to find a random spot in the genome that is similar to it. The computational cost of doing this is high enough that for multicellular genomes, you don't do it on your laptop. If you're not in one of the sort of comparative genomics labs, you probably don't have in-house hardware to do that. So we'll do it on bridges just to show you how to do it on bridges. But the other thing is it benefits a lot from running in parallel, right? Because if you've got millions and millions of these little sequences to try to match to the genome, the more cores, right, you're essentially multiplying the speed by however many cores you can use, because each of those cores can be looking at the same time. They don't depend on each other. Okay, so the first step, though, sequencers are imperfect. No library prep method is perfect, and so we get contamination. We get some of those adapters I was talking about that tether it to the genome or to the flow cell, the glass slide. All of those things are things that we want to get rid of if the objective is to call variants, right? We don't want to say there's a variant in there when really it was contamination or a sequencing error, especially if you're talking about clinical sequencing. So the one that we've used in the past is called FastQC. This is a good tool if quality control is your objective. And then you could use trimming software downstream of that. We're going to actually use a new software called FastP, which does both the quality control assessment and the trimming all in one. Before we jump into it, let's actually get on the data, get on bridges. So go ahead and upload the data from Canvas to your bridges home directory. Right, so the data for the tutorial is here on Canvas. You're going to move that, excuse me, onto Bridges 2, and then unpack it. I've given you the code to unpack here. And fair warning, copy pasting from the PowerPoint slides is hit or miss. If I were you, I would try copy pasting from the PowerPoint slide, but if you get a weird command not found or something, then you might have to type it. The main ones that trip people up are sometimes it pastes the wrong kind of dash, like it'll paste in an EM dash instead of a regular dash. Sometimes it'll put a invisible tab character that's not the normal one. I don't know why these things happen. These are just experience. Especially when it's invisible characters, there's nothing more frustrating. Oops. Yep. Yep. Um, in a minute, you'll have to start an interact session. You don't need to be in an interact to unpack a file. My directory's got a bunch of extra stuff because it's already run the pipeline, but you should, in your directory, just have the GVA data tar.gz. And I guess making a directory, if you already have a bunch of stuff in your home directory, then maybe creating a new directory and putting this in there, because you're going to generate a lot of files here. Well, a lot, 10 or 20. Sort of like the BLAST project. The genomes need to be indexed, so that generates files. And then when it's mapping reads, it does it too. Calling variants. So once you've run that tar command, actually, so where are we with the tar command? How many people are done with the tar command? Half-ish. No? Out of storage space where? On your computer or on? Oh. It's not that big. It's 100 and something megabytes. I don't have a quick and easy solution for that one. I keep losing my window. There we go. Yeah, the whole archive is 270 megabytes. Okay. Hmm? How many of you are in genomics? Did you guys get to use Galaxy today? Well, no, yeah, I asked about today specifically because yesterday it was completely down. Matt was having a minor crisis. It's up right now. I was just thinking that Bridges crashing is way more rare than Galaxy crashing. Okay, so first task, look at one of those FASTQ files. Now, the FASTQ files are still zipped. If you were to use the gzip command to unzip those, they're about double to triple the size. So don't need to do that to look at them. There are these handy tools called zless or zcat, and it says look inside that zipped file and report. So just like you would use less or cat to look at any other file, you can look into those. Now, if you use less or cat, these files have millions of lines. I forget how many exactly. So you want to pipe that into head, right? So let's do this. All right, so we can do zless srr tab 1 tab. So that's saying use the Z to go use less in this gzipped archive. And then I'm going to use pipe head 4 because each sequence in a FASTQ is 4 lines long. Okay. Oh, what'd I do wrong? Forgot the little dash before my 4. Okay. I'm going to do the same thing on the read 2. And so one thing to notice here, this is the format for all FASTQ files. So what you get is the header, which is the name of this read. Okay, that's that line right there. Then you get the read itself. So those are the base calls. And this happens on the machine. So I told you the raw data for a Illumina sequence is actually the images. Those images get converted into sequence on the machine. Okay, so those images do get saved. You can go back to them if there's a problem with the flow cell, or example. There are ways, especially usually this is contacting Illumina to go back and look at it. The next line is always, in my experience, just a plus sign. It's left there for comments, but I never see comments. Okay, at least I think that's what it's for. And then the fourth line is the quality score for each base pair. So there's one character that corresponds to each one of the base calls, and each character, like that A right there, isn't interpreted as the letter A. It's interpreted as the ASCII value. So it actually has a numerical conversion to say how confident are they in that base call. So that conversion that happens between the image and calling that first position a T gets a score for how confident they are that the image corresponds to a T. And then that confidence is that bottom line there. So the semicolons and the question marks, it kind of looks like a gibberish string, but it's really telling you the quality of each score. Now, our quality control software, the FastQ or FastP, knows how to read those as quality metrics. And so that's how it's going to interpret the quality of the reads to know whether these are any good. Because you can imagine, if it's a really low quality base call, you don't want to tell somebody they've got a mutation there if it's just a sequencing error or just a base calling error. Okay. The other thing to notice is this is the first sequence from read 1 file, and this down here is the first sequence from the read 2 file. The only thing different in those two lines is the last character of the read name, right? 1 versus 2. So that's how the software knows to keep, these are paired ends that belong to each other, okay? Go back to the PowerPoint. So that was looking at the first read. If we wanted to look at the last read, what would you do? I'm not going to actually do it, but yeah, instead of doing head 4, you could do tail 4, right? And that will give you the last 4 reads in the file. If we want to know how many reads are in the file, I've given you a pretty strong hint there. zgrep works on zipped files. The dash, so normally if you want to count, grep-c is to count whatever the pattern you give it is. So what pattern could we grep that will tell us how many sequences are in these files? So in this line, replace search string with something that is going to tell us how many. If it's a FASTA file, what do we usually do? Grep the greater than sign, and that will tell you how many FASTA headers are in there. That tells you how many sequences are in your file. That's the answer to the first 6 quiz questions, right? For this, like the blastome, like how many proteins are in this file? Grep the greater than sign. That tells you how many proteins are in there. Okay. You can't do that for FASTQ files because they don't all start with the greater than sign. Okay. So what might you grep? So I'll go back here where I've got exactly like this, but they're very similar. What could we search? Okay, that's a good guess. The way it might go wrong is if at is actually like its ASCII character can be used as a quality score metric. So it could show up in the fourth line in the quality scores. But that's definitely the right track. If we extend that to @srr, which every sequence starts with, because it uses the file name, that is certain not to match, because all 4 of those are, you could use the whole file name. Right? So searching on, whoops, zgrep will answer this question. Actually, let's try it your way and see if it comes up with a different number, because that will answer the question. Oops, I forgot dash C. So 3,813,000 sequences reads, okay? That's if we just, I'm just curious. I wonder if they actually don't remember the answer to this. No, okay. So that tells you how many times at is used in a quality score, something like 13,000 times. So really only 3,800,180 reads. Okay. So that's number of reads. That was just to count for read one, but you should find exactly the same number. If you ever have data sets where they're not the same, that's a problem. And it will make the aligners, most of them, it will make them die. Okay, so you'll get an error. It'll say not all the reads have pairs. All right, are the sequences any good? So this is what I was saying, that up until a few years ago, you had to use FastQC to find out if they were any good, look at them, and then go back and use a read trimmer based on the quality information you got from FastQC. FastP has that all in one, so it will do the analysis and provide the outputs for you. I've given you links to FastQC because it's got a really good sort of explanation for why you would or wouldn't trim reads. And these links, so the quality scores, oops, you can't see what I see. Okay, so I did that, the good Illumina data. This is sort of the per base sequence quality for a good sequencing run. And so what it's showing you is the quality score on the y-axis. So again, those are that last row that had A's and semicolons and question marks. They all translate into a number from 0 to 40, maybe 0 to 38. 38 is as high as it goes on this graph. But you can see that no matter where you are, the x-axis here is the position in the read. So these are whatever the pretend data is here is 40 base pairs. And almost all the way across the read, the average across all reads is very high score. So the base calls, you're very confident in base calls all the way across. Whoops. Contrast that with a bad run, it will look something like that. So in this case, what you can see is down here at the tail end, so those last few bases, they fall below, like they're out of the green zone. So usually we would say if it's below 30, especially in human genetics, if it's below 30, throw it away, because we don't want to risk calling that a true variant if we're not confident in the base call. And so this is where you would say, if I had a run like this, I might say, okay, we need to trim off the last 10, 14 base pairs off of this read. Or some trimmers will say, as you go along the sequence, when the average goes below, say, 30, then trim everything downstream of that. So it can trim in windows. There are different algorithms for that. FastP is like the new standard. Every paper I see uses FastP now. And most of them don't tune anything. It's just enough people have done this enough times that they just wrote the software to do it all for you. But this is what it's doing. It's analyzing those quality scores and deciding this is what we should keep and this is what we should not. This pattern is super common. So even in the good sequence data, you see how it's worse at the end. That is all Illumina reads do that. It's just that the sequence always degrades the more cycles you run. That's what limits the length of the reads. So why Illumina sequencing technology only gives you like 150 base pair reads, or now you can get up to 300, but it's because their enzymes, their fluorescent markers, they degrade the more times you shoot them with the laser. So, okay, we're out of time, so we'll pick up there. Yeah, if you want to try it on your own before Thursday, I would say you can try this next slide, okay, which we've done before, but this is an interact session, so this will run FastP. You're going to load the module Anaconda. You've got to do that so that you can import FastP. FastP is not on bridges, so you've got to install it. The only way to do that is through those first two commands. Then you've got to activate that environment that you create so that the interact session will be able to run FastP. And just so you know how to read this, those backslashes, what do? I've asked this one before. It says don't execute this line yet. The lines are continuing. So really you could type all this in on one line and leave the backslashes out. I just wrote it that way for clarity. All right, we're out of time. So we'll pick up with QC there and get through variant calling on Thursday.